So we talked about the fact that between glycolysis and the Krebs cycle, we have the modification and the chemical modification of pyruvate in order to cause it to become acetyl-CoA. We didn't actually talk about this specific process and how it actually occurs. So under aerobic conditions, when you have the mitochondria inside a cell, you are required to take pyruvate and to chemically modify it into create a molecule called acetyl-CoA. This process is basically occurring on the inner membrane of the mitochondria in a specific structure called pyruvate dehydrogenase. It is when we take the pyruvate and through a specific sequence of processes that are basically going to occur in this pyruvate dehydrogenase, we are going to create acetyl-CoA. And acetyl-CoA is actually the first thing that's going to enter the Krebs cycle, which is occurring inside the matrix of the mitochondria, which is going to keep the cellular respiration. But for now, let's focus on this specific reaction, the link reaction that links between glycolysis and the Krebs cycle. So pyruvate enters this structure, this combination of a few structures, and it is going through a couple of steps. It is going to be decarboxylated, which means that the pyruvate is going to lose a carbon atom. It is going to lose a carbon dioxide to the environment. 2, we are going to oxidize the pyruvate and we are going to reduce NAD+ to NADH, which means, and the name suggests, we are going to dehydrogenate the pyruvate. We are going to remove hydrogen atoms and we are going to reduce NAD+ to NADH. We are also going to insert a very important and very special molecule called coenzyme A. Coenzyme A is quite complex molecule which you don't need to know the structure of, but this molecule is going to combine with the acetate that we form from the reduction, from the oxidation and the decarboxylation of pyruvate, and we are going to get acetate, which is going to be combined with CoA, and it is going to give us acetyl-CoA. Just a combination of acetate and coenzyme A. And this is how you actually link the glycolysis and the Krebs cycle that we are going to study. The thing is that it is very high yield for the IMAT and BMAT exam. And when it comes to the IMAT and BMAT exam, they are going to expect you to know what are the products of this reaction from pyruvate and acetyl-CoA, from one single glucose molecule or from one single pyruvate. So let me give you an example. One glucose molecule is going to produce 2 acetyl-CoA. Why? Because one glucose molecule is going to produce 2 pyruvate, and 2 pyruvates are going to go, each one of them is going to do the link reaction once. So the link reactions are going to occur twice for one glucose molecule, which means that while you have one carbon dioxide and while we have one NADH, for one glucose molecule you are going to have 2 carbon dioxide and 2 NADH because you have 2 pyruvates, which is so important to know and memorize for the IMAT and BMAT exams. So don't confuse between the products of one pyruvate or the products of one glucose when it comes to glycolysis, link reaction, and in the future where you study about the Krebs cycle itself. I hope this video made sense. I hope it helped you to understand the process of the production of acetyl-CoA from pyruvate through this special process and chemical modifications we are doing to the pyruvate in order to prepare the next substrate we are going to use inside the Krebs cycle.